Hello, this is the video for the presentation of poster entitled Using Metaheuristic Algorithms to Analyze Small Angle Scattering Patterns for the symposium B9 Nutrients in Nanoscience and Nanotechnology, Innovative Synthesis, Novel Properties, Theory and Challenges at the 31st International Materials Research Congress 2023. I am Diego Ramirez, a physics engineering student and researcher at the University of Cauca in Colombia. Together with my colleague, Estibel Camayo, also a student and researcher, the PhD Diego Coral and the PhD Carlos Cobos, we are conducting research that focuses on evaluating the performance of the computational reverse engineering analysis for scattering experiments CREASE method under modification in its algorithm. CREASE is a method recently developed by the University of Delaware, capable of obtaining key structural information of the shape factor and the structure factor from the small angle scattering SAS profile of a sample, and even making a representative 3D structure of the sample, useful for further analysis. It is capable of analyzing different types of samples, such as solution of mixtures of low and high concentration nanoparticles, and is easily adaptable for new sample types. Small angle scattering is a group of well-established standard techniques useful for studying the structure of matter and its interactions, especially used for analysis of non-periodic structures of colloidal size with scales from about 10 angstroms to several thousand angstroms. Among some of its applications is the characterization of colloidal particles in solution, as well as solids. In SAS experiments, a primary beam of radiation, usually X-ray or neutrons, is incident on the object of study, elastically scattering the radiation and producing a 2D scattering pattern. In the case of isotropic samples, a 1D pattern can be obtained by radial averaging. Here are some examples of SAS patterns from different samples. The success of SAS lies in the ability to choose the appropriate mathematical model that allows obtaining physical information about the analyzed system from the SAS pattern. Then, what is CREASE? CREASE, or Computational Reverse Engineering Analysis for Scattering Experiments, is a method for analysis of SAS patterns. CREASE is based on the rigid body analysis technique, which consists of modeling the sample or part of it as a conformation of subunits, the rigid bodies, with known structure, and using movements of these to find the spatial arrangement whose SAS profile best fits the experimental one. The CREASE method uses descriptors from which it locates the subunits. For example, in the case of a solution of low concentration vesicles, its descriptors are the radius, the thickness of their layers, and the dispersion of these dimensions, among others. With this modification, the dimensionality of the optimization problem is lowered, and therefore its computational cost is reduced. The CREASE takes as input the experimental SAS pattern of the sample, through an internal optimization using an adaptive genetic algorithm AGA, a metaheuristic based on Darwin's evolutionary theory, to get key structural features of the sample, which we called descriptors. Then, how works the CREASE method? The CREASE begins generating a group of combination of random values of descriptors, called a population in the AGA. Each combination is called an individual. Then the SAS pattern is computed for each individual, and the fit of the calculated SAS patterns is evaluated with the experimental one. From this, using the genetic operators of the AGA, a new population is generated, and the above process is iteratively repeated to finally get a combination of descriptors whose SAS pattern is closest to the experimental one. Also, a representative 3D structure of the sample, useful for further analysis. The calculation of the SAS profile of an individual is done by locating rigid bodies based on its descriptors, and with this, from the divide formulation, it is possible to obtain the computed SAS profile corresponding to that individual. This method requires taking the distance between pairs of scatterers, and this results on a high computational cost due to the large number of arithmetic calculations needed. Although versions have been implemented in which the use of the divide formulation is replaced with the use of an artificial neural network, that allows for lowering the computational expense. The function used to evaluate the fit of the computed SAS profile with respect to the experimental one is the metric sum of squares of error, is E, but another metrics could be used. The objective of our research is to evaluate the efficiency of an alternative optimization algorithm to the AGA used in CREASE, aimed at improving the computational efficiency of the method. Since it currently requires considerable computational resource to work. For example, for the amphiphilic polymers in self-assembled vesicle research, the CREASE team used the high-performance computing resources of the Faber supercomputing clusters at the University of Delaware. In the test we performed on Intel Core i7 computers, we found that the machine times required for the execution of the method are around 12 hours. It should be noted that most of the processing and machine time is used in the calculations of SAS profiles. Therefore, we seek to reduce the number of computed SAS profiles necessary to achieve the most optimal combination of descriptors. This can be achieved by improving the optimization algorithm used. After an exhaustive literature review, it was decided to evaluate the global best harmony search GHS algorithm as alternative to the AGA. The GHS is a metaheuristic inspired by the music improvisation process. To perform this evaluation, we will run the CREASE method with the AGA and GHS on 4 benchmark functions, which are SAS patterns of samples of low concentration vesicles solution with known dimensions. For each combination of optimization algorithm and benchmark, 30 repetitions of the experiment will be performed. The results of these runs will be used to evaluate the performance of the CREASE with each algorithm using no parametric test. Preliminary tests of the use of the CREASE with the GHS algorithm have been performed using these hyperparameters. For each CREASE run with AGA and GHS, 8,000 SAS patterns were computed in total. This on this in silico benchmark of a low concentration solution of amphiphilic polymers vesicles of known dimensions, an inner radius of 10 nanometers with a log normal dispersion of 20%, a thickness of solvophilic layers inner and outer of 12 nanometers, and a thickness of the solvophilic layer, the intermediate one, of 6 nanometers. The results of the test were as follows. Here we can see the evolution of the best SSE during different CREASE runs with the AGA and the GHS. We can see how the GHS shows a higher convergence speed than the AGA, reaching a stable SSE value faster and more uniform between runs, unlike the AGA that between runs shows a higher variance between the results. Here is how the best computed SAS profile evolves during a complete CREASE run for the AGA and the GHS. It can be seen that the GHS achieves a computed dispersion profile very close to the experimental one, as well as the AGA, even improving the fit for some Q values as in this area here. Finally, the vesicle descriptors obtained from these runs were the following. It can be seen that the GHS achieves descriptors closer to the SPED ones with a lower variance, with respect to those obtained with the AGA. This allows us to conclude preliminarily that it is indeed possible to improve the performance of the CREASE method. In this benchmark used, the GHS shows a better performance equaling and improving the results achieved by the AGA, even though it is necessary to test in other benchmark. On the other hand, the GHS hyperparameters can be adjusted to further improve the performance of CREASE with GHS, increasing the convergence speed, precision, and accuracy of the obtained parameters. In the future, we wish to do this efficiency evaluation on CREASE, which can obtain information about the form factor and structure factor simultaneously for samples with high concentrations. Finally, we want to acknowledge Professor Arti Jairaman's research lab at the University of Delaware in Newark for the support in the use of its method CREASE, and the University of Cauca. And we want to thank you for your attention. For any communication, questions, or suggestions, you can contact us through the email shown on the screen.